So in this section, we're going to cover gene mutations. So gene mutations is a permanent change in the sequence of bases. And you can imagine that if there was an effect or sort of a change in the DNA, it could have some effect on the protein. Because we had learned through gene expression, we start off with the DNA, it gets transcribed through transcription into mRNA, messenger RNA. Then that messenger RNA, through the process of translation, gets read into the polypeptide or protein. So if there is something that happened at the DNA point, it could have effects at the end. And so if you look here at this figure here on the left side, how mutations alter a genome, again, I just want to reiterate that if you have a mutated DNA, then that can result in a mutated mRNA. That mRNA can alter a protein's function, alter a protein, that 3D structure, or it can alter its function. So when we're talking about gene mutations, there's a range of effects that could happen. Range of effects would be that, for one, it could change the expression of the gene, or it can be such a huge change that it completely just inactivates the protein. There are a couple different causes of mutations, these different causes of mutations. One is going to be spontaneous mutations. So these arise due to the abnormalities in normal biological processes. So in other words, they just kind of happen. Then there's another cause, which is called induced mutations. So thinking of the word induce, it means like to start. So these are started most generally by environmental influences. So examples would be exposure to radiation, even just being outside, those UV rays are hitting your skin. That can cause mutations, or carcinogens. Those are other sort of toxins that can cause mutations. And then there's those that are caused by errors in DNA replication. So usually these are pretty rare. And the reason it's rare is that there's the DNA polymerase. We have learned that it makes the copy of DNA. So DNA polymerase makes DNA in DNA replication. But DNA polymerase also has another function. So we learned that the function when we were talking about DNA replication was that it reads that template strand and it gives complementary base pairs. But sometimes it makes a mistake. So it has this proofreading job also that's like, oh, I was supposed to add a T here, and instead I put a G. So I'm going to go back, take that T out, and I'm going to add the G. So usually DNA polymerase is able to fix that before it is then made into mRNA. Well, there's a couple different types of mutations, and two of which I'm going to talk here. So the first type of mutation would be point mutations. So point mutations are a change in a single DNA nucleotide. So for example, here we see our normal strand of DNA. This is also called the wild type. And then we have our mutant strand. In this case, I'm calling it the point mutation. And you can see there's this change, a single change in the nucleotide. So this change is going to be where the C was changed to a G. The next type of mutation is a frameshift mutation. So this is where one or more nucleotides are either deleted or inserted from the DNA. So the result can be that it can cause a completely new sequence of codons, or it can cause a non-functional protein. So for example of a deletion, so if we're looking at our normal strand here, so this is going to be our normal. If we're looking at our normal strand, we have our AGA, C, C, G, C, G, GCC. And this codes to arginine, proline, alanine. But let's say in this deletion that these two base pairs here get deleted. So the sign here means it gets deleted. When that gets deleted, it changes. It changes our nucleotides. So we have AGA as our first three, and then we end up with our CGCC, which comes from our normal above. So we have our CGCC. So this deletion causes a change in frame. So remember when we were reading nucleotides for mRNA, every codon is a codon of 3. But if you delete 2 of those, as we had shown in this example, then it shifts that frame. And in this case, it actually caused a change from that proline to an arginine. Opposite of that could happen. An opposite thing could happen where there's an insertion of nucleotides. So here, if we go back to our normal, we have our arginine, proline, alanine as our normal sequence or wild type sequence. And here, right after the AGA, we get an insertion, then that shows here. So we could get an insertion right after this A of a CC. So when there's an insertion after that A of CC, we end up with a sequence at the bottom. So we have our AGA, then we have our CCC, which would be these guys, that's 1, and then we have this as our next codon, CGG, and then CC at the end. So at this point, our frame changes, and that's what you see here. Okay. The third type of mutation we're going to talk about is a silent mutation. So silent mutation is where a resulting protein has the same amino acid sequence as before the mutation. So for instance, again, we have our normal, and let's say we have this point mutation from a G to a C, as you can see here. We go from a proline CCG to a proline CCC. So even though this third nucleotide of the second codon changed, it didn't have an effect. And you can see that based off of the codon chart. So here on the right, we have our original proline from our normal strand as being this CCG, and then we get that to change to a C here. So there's our chain C, but as a result, it's still a proline. It also turns out by looking at that chart that if it had changed to a U or even an A, it still would be proline. So in this case, the amino acid would still be there. The correct amino acid would still be at that point. So the silent mutation, again, is where a resulting protein has the same amino acid sequence as before the mutation. The next type of mutation we're going to talk about is going to be the missense mutation. And this is where a mutation where the resulting protein has one different amino acid than before the mutation. So it changed it. It changed that amino acid. So here's our normal. Oops, wrong one. Here's our normal. And from our normal, we're looking at our AGA, and then there's this point mutation of a C in the first codon. Oh, we have a CCG, and then there's a point mutation here where the C gets changed to an A. When that C gets changed to an A, it causes this proline and the normal to get changed to a threonine. So here we would have our, if you look at our sequence here, we have our proline, which we have a CCG on our case up here, so it's going to be this guy. Oh, shoot. Sorry, my cat is like all over me right now. You shifted the screen and we were doing so well. Okay, let me get back to where I was at. So we have our proline in our example here for missense mutation as being a CCG. So this is our CCG proline. And then that gets changed to an A. So now we have this A that gets changed with this sequence. And then when that happens, it gives us a threonine. So then this is a missense mutation. Completely changes the amino acid, in this case from a proline to a threonine. Then the last type of mutation I want to bring up is going to be a nonsense mutation. And this is where a mutation occurs where a stop codon results from the mutation. So we have our normal sequence AGA, CCG, GCC, and our mutation is going to change this codon, the second codon from our proline, and it's going to change it to the stop codon. So here, if we go back to our proline example, we have our CCG. This is our proline example. And then the two base pairs in the second codon get changed to a UA, and that becomes a UAG here. So then that causes it to become that stop codon. So when you're looking back at the different mutations, there's 5. So we have our point mutation, which is going to be a single change in a nucleotide. So if you're looking at the word, a point is one nucleotide gets changed. It can be 2 sometimes. Then we have our frame shift mutation. So if you're looking at the frame shift name, it's shifting in frames of codon. So one deletion or 2 deletions can cause it to shift frame, or insertions would cause it to shift frame too. A silent mutation results in not a change in the amino acid. So that even though it's a change of the nucleotide, the amino acid is still the same. And then missense mutation and nonsense mutation. So missense mutation is where a resulting protein is a different one. So missense is a different one, and then nonsense is where there's a stop. So there's 2 things. There's at least one way that it helps me remember the stop codon. So when I'm thinking of the stop or the nonsense one, I always write, at least with this one, I write in the name a stop sign where the O would be. So this would be nonsense mutation. And missense is just a change. So I think, I just sort of think that the other one's a change of it. But nonsense for sure, I know it's going to be the stop codon, so missense would be the other one. That's just one way that helps me remember it. Just because those 2 names sound very similar, missense and nonsense. So the last part of covering, the last part for this section would be to complete the class activity. To complete the class activity of mutations, I have that attached to your remote page. And then when you're done with that, I have the key listed at the end here. So here is going to be what the class activity looks like. You're doing a review, sort of review of what happens with gene expression. And then on the second part, what you'll do is look through to see what type of mutations would happen based off of an amino acid there. So it's a good way to practice just thinking about how to read nucleotides and then going into seeing whether they will cause a mutation in that.